Hello everyone, I Kulpreet Kaur. Today I am going to prepare an audio record for visually impaired student on the topic Bioinorganic from Pradeep. Pradeep Inorganic Chemistry Volume 3 by Dr. K. Bhasin. Now let's start the topic Bioinorganic Chemistry. Inorganic chemistry deals primarily with salts and complex metal ions, whereas bioinorganic chemistry deals majorly with inorganic chemistry when applied to biological system. Thus, bioinorganic chemistry can be regarded as a branch of science that relates inorganic chemistry with life processes by recognizing the role of metal ion in the normal growth and function of biological processes. In brief, bioinorganic chemistry highlight role of metal ions in biology. Bioinorganic chemistry is a rapidly developing area of chemistry because of its importance in biological system. The essential biological roles of a few elements have been known for many years. This chapter will focus on the important role of metals, nonmetals, and their compounds in biological processes, essential and the trace element. We think that chemistry of life is slowly composed of organic molecules. It is a wrong concept. In fact, there is a long list of inorganic compounds that are essential to living system. For example, the skeletal and the integumental materials in higher animals is composed majorly of inorganic materials, and the lifetime consumption of oxygen by a living entity is a major fraction of its material need. In addition, water, oxygen, calcium phosphate, and calcium carbonate are the major inorganic constituents of living matter in terms of quantity. Table lists the abundance of elements in earth crust and in ocean compared with some abundances in typical mammals. Of all the known element, it is about 30 elements that have been recognized as essential and indispensable of life. Of the 30 elements, 15 of them metals. The essential elements are needed for the growth and normal function of the animals and plants. Absence of these elements result in severe malfunctioning of the living organisms. Thus, an element may be considered essential when its insufficient intake produces an impairment of normal function, and its restoration relieves the impaired function. Some of these elements are required in bulk or microscopic amounts to all forms of life and are classified as bulk element. Some of the bulk elements are required for structural purposes: skin, bones, eggshell, various organs, while the rest are used as minerals. In other words, in the absence of essential elements, the organisms can neither grow nor complete its full lifestyle cycle. The essential elements can be further subdivided into two groups: bulk elements, trace element. And the bulk element is essential element that are formed in higher concentration. Trace element, essential elements that are present in low concentration. Bulk quantities of group 1 and 2 metals are required mainly to balance the electrical charges associated with negatively charged macromolecules in the cell, and also to maintain the osmotic pressure inside the cell. The reaction flask of living organism is the cell. In addition, the transition elements and the heavier members of the P group also play an important role in the bioinorganic chemistry. Some of the inorganic elements are required in small quantities and are classified as traces or ultra traces elements. These different types of essential elements present in living system are classified in table. Bulk structure elements: hydrogen, carbon, nitrogen, oxygen, phosphorus, sulfur. Mineral elements: sodium, potassium, magnesium, calcium, chlorine. Trace elements: iron, copper, zinc, vanadium, nickel, cadmium, lithium, lead, tin. Metal, ultra trace element, metal, manganese, molybdenum, chromium, silicon, arsenic, selenium. Non metals, fluorine, iodine, boron. It is important to note that not all the elements displayed in table are required by all the plants and animals. For example, sodium ion Na positive is of special importance to higher animals. It is, however, not essential to many bacteria and plants except blue-green algae. The ultra trace elements boron, aluminium, and vanadium are needed by some higher plants. Molybdenum is essential to only those organisms which derive their nitrogen from inorganic sources. Selenium is an essential micronutrient for mammalian species but proves to be toxic at higher levels. Selenium deficiency in human results in a degenerative condition of heart tissues known as Cation disease, in which it was endemic because of extremely low selenium levels in the soil coupled with a population that entirely depended upon locally grown grains. It is important to note that even some essential elements may prove to be toxic if consumed in larger quantities. Next topic, essential bulk element, sodium and potassium. We have learned that both sodium and potassium are essential for life processes. For example, we need at least 1 gram of sodium ion per day in our diet. However, because of our habits to adding salts on food, some people consume as many as 5 times more than required. However, intake of potassium is not a problem. Instead, potassium deficiency is more common. We must include potassium rich food such as bananas and coffee in our diet. In organic chemistry, we always think of the similarities between sodium and potassium. However, in biology, it is the difference between sodium and potassium which plays important role. Cells pump out sodium ions of the cytoplasm whereas the cells pump the potassium ion inside. This ion transport is sometimes called as sodium pump, and it involves both the active expulsion of Na positive ion and the active take up of K positive. It is the difference in the total alkali metal ion concentration inside and outside the cells that produces an electrical potential across the cell membrane, which is important for the functioning of nerve and muscle cells. Concentration of ions. Ion रेड ब्लड सेल्स Na पॉजिटिव कितना 11, K पॉजिटिव 92. Blood plasma Na positive 160, K positive 10. The transport of iron require energy, and this is obtained by the hydrolysis of ATP, adenosine triphosphate. It is estimated that hydrolysis of an ATM molecule to ADP, adenosine diphosphate, provides enough energy to move 3 Na positive ions out of the cell and 2 K positive and 1 H positive ion back into the cell. Calcium and magnesium. Both calcium and magnesium are present in the body fluids. Mg2 positive ions are concentrated in the animal cells and Ca2 positive ions are concentrated outside the cell. This is similar to the presence of K positive ion inside the cell whereas a Na positive ion outside the cell. Ca2 positive ion are present in the external skeleton of creatures such as shellfish and corals. The material used is calcium carbonate. Creatures with interior skeleton such as mammals and reptiles use calcium hydroxide phosphate, Ca5OH, PO43, and the enamel on teeth as fluoropatite. 3Ca3PO42 CaF2. Both these compounds are highly insoluble. Important minerals used in biological materials are listed. Ca2 positive ions are important in blood clotting and are required to trigger the contraction of muscles such as those that control the beating of the heart. In fact, Some types of muscle cramps can be prevented by increasing the intake of calcium ions. Calcium containing minerals in biological materials. Mineral calcite formula CaCO3 organism birds location eggshells. Ergonotite formula CaCO3 organism molluscus shell. Hydroxypetyl Ca5PO43OH organism vertebrates location bone. It decreases Ca2 positive content of blood either due to dietary deficiency or from an sufficiency of the parathyroid hormone may lead to a disease called TNT. On the other hand, an increase in the concentration of Ca2 positive ion in the blood may lead to calification of several integral organisms. The absorption of dietary Ca2 positive ions from the intestinal tract may be prevented by the simultaneous ingestion of organic acid, example oxalic acid, with it combines to form an insoluble salt. It means that availability of dietary Ca2 positive is greatly affected by the other constituent of diet. Both calcium 2 positive and magnesium 2 positive ions are excreted mainly via the large intestine instead of the kidney. Magnesium, because of its high charge radius ratio and consequent strong hydration, as in magnesium H2O6 2 positive, plays important biological roles that are different from those of calcium. It acts as counter ion to the negatively charged group in nucleotides and polynucleotides. One of the most important nucleotides is ATP, adenosine triphosphate, which serves as the fundamental energy source for the living cells. ATP is thus known as energy rich molecule and is used to drive most of the chemical reaction of the cell. Mg2 positive ions form a complex with ATP. It has been observed that all transphosphorylation reaction involving ATP proceeds smoothly in the presence of magnesium 2 positive. ATP is an anhydride of phosphoric acid. The hydrolysis of anhydride is a very favorable reaction. Thus, the hydrolysis of ATP to ADP shown in the equation below liberates 30.7 kJ per mole of energy at pH 7. The living system thus utilizes this energy to drive energy requiring biochemical processes. It has been observed experimentally that the activating effect of Mg2 positive ion transphosphorylation may be duplicated in vitro by magnesium 2, Ca2 positive, cobalt 2 positive, zinc 2 positive, or nickel 2 positive. The most important aspect of the biochemistry of magnesium is its role in photosynthesis. Magnesium containing chlorophyll using energy from the sun converts carbon dioxide and water into sugar and oxygen. 6 carbon dioxide 6H2O सी क्या बनाते हैं C6H12O6 + 6O2. Without the oxygen from the chlorophyll reaction, this planet would still be surrounded by a dense layer of carbon dioxide. In addition, without the sugar energy source, it would have been difficulty for life to progress from plants to animal. It is important to note that magnesium ion sits in the middle of the chlorophyll molecule and holds the molecule in a specific configuration. Magnesium has only one possible oxidation number, positive 2. As a result, the electron transfer reaction involved in photosynthesis can proceed without interference from the metal ion. Phosphate. Phosphorus is an important element for life processes. For example, free hydrogen phosphate PO43 negative and dihydrogen phosphate H2 PO for negative ions are involved in the blood buffering system. Phosphate is the linking unit in the sugar esters of DNA, deoxyribonucleic acid, and RNA. The phosphate unit make up part of ATP, the essential energy storage unit in living organism. Bone is a strong phosphate mineral. The commonly called mineral apatite is calcium hydroxide phosphate. Ca5OH, PO43. The presence of phosphorus in bonds and its function in regulation of pH of blood constitute important features of phosphates. It is important to mention that the normal formation of bonds depend upon the relative amount of dietary phosphorus and calcium in addition to vitamin D. Essential trace elements. Among the non-trace elements, the three most prominent biologically active metals are iron, copper, and zinc. The remaining trace elements, excepting boron and fluorine, are used in very small amount than 10 mg in adult human and are referred as ultra trace element. The trace elements are known to play important role as metal enzymes. Iron is essential in small amount for both plants and animals like life. However, like copper and selenium, it is toxic when used in larger quantities. Biologically, iron is the most important transition element and is found in greatest abundance in biological system in the form of porphyrin complexes or hemes such as hemoglobin and myoglobin. Porphyrins are microcyclic ligands and are derivatives of porphyrin discussed later. A human body weighing about 70 kg contains 4.3 gram of iron. Nearly 70% of this iron is present in hemoglobin and about 3% in myoglobin. The remaining 23% body iron is present as ferritin. Ferritin is present in spleen, liver, and bone marrow. It consists of a shell of linked amino acid called apoferritin surrounding of a core of an iron 3 oxohydroxophosphate. The core is a cluster of iron 3 ions, oxide ion, hydroxide ion, and phosphate ion, and the composition FeOOH8, FeOPO3H2. It is very large, containing up to 4500 iron ions. With its hydrophilic coatings, this large aggregate is water soluble and is concentrated in the spleen, liver, and bone marrow. Apoferritin protein can be prepared from ferritin by reduction of ferric ions to ferrous ions, pH कितना है इसका 4.5, followed by the removal of Fe2 positive by dialysis. The formula weight of apoferritin is nearly 45,500. Ferritin can also be synthesized in the laboratory with properties similar to natural ferritin. It can be prepared by treating crystalline apoferritin with an inorganic Fe2 salt in the presence of oxygen. Ferritin is probably formed in vivo by analogous process. The various steps involved in the pathway Fe2 positive from the food stuff to hemoglobin and to ferritin involves the following processes. The Fe2 positive of the bound dietary material is reduced to Fe2 positive in the gastrointestinal tract. After absorption into the cells of intestinal mucosa, Fe2 positive is incorporated into ferritin as Fe3 positive. The Fe2 positive in mucosa is also converted Fe3 positive plasma bounded by iron binding globulin named transferrin. Plasma Fe3 positive is in equilibrium with iron in the liver, spleen, and bone marrow. The following changes that occurred are summarized in the scheme. Bound Fe3 positive transfer in porphyrin serum Fe2 positive, liver, spleen, bone marrow, ferrin Fe3 positive, opoferritin, globin, hemoglobin. Pathway of iron 3 from the food stuck to hemoglobin and to ferritin. Zinc. Zinc. Among the trace essential elements, zinc is second only to iron is importance. Zinc is essential to all organisms and plays an important biological role in the enzyme system of animal and plants. Human contain about 2 gram of zinc and is one of the most important and crucial elements in our diet. Deficiencies of zinc are not life threatening but contribute to fatigue, lethargy, and related symptoms. Zinc 2 positive ion is a soft acceptor compared to other divalent ions. Mg2 positive, calcium 2 positive, or Mn2 positive and act as a stronger Lewis acid towards many biomolecules compared to zinc 2 positive ion. Cu2 positive ion is also soft but has an additional intermediary oxidation state, that is Cu positive. Different oxidation state of copper may allow different redox reaction and complicate its behavior. The complexes of zinc ion are much more liable than the corresponding complexes Mg2 positive, nickel 2 positive, or Mn2 positive. These facilitates the role of zinc ion in enzymes. Since zinc 2 positive ion is completely resistant to redox changes at biological potential, its role cannot be affected by changing the redox potential in the organisms. A number of enzymes containing zinc ion are known. The most important of all the enzymes is the carbonic anhydride. It is present in red blood cells and is involved in the respiration. It speeds up the absorption of carbon dioxide by red blood cells in muscles and other tissues. On the other hand, in the reverse reaction, it involves the release of CO2 in the lungs and reaction is pH dependent. CO2 plus H2O, carbonic anhydride H2CO3. Thus, the zinc containing enzymes carbonic anhydrase catalysis the otherwise show carbonic acid carbon dioxide equilibrium reaction. The carbonic anhydrase has a common for coordinated environment around a Zn2 positive ion in which three of the ligands are imidazole nitrogens of 3 histidine and the fourth is a water molecule or hydroxide ion as shown below. A water molecule coordinated to a zinc ions has pK near 7 and thus the role of zinc is to reduce the basicity of hydroxide ion such that a high effective concentration of hydroxide can exist in an approximately neutral medium. Another important enzyme containing zinc 2 positive ion is carboxypeptidase. It is present in the pancreatic juice and is primarily involved in the digestion of protein by animals and protein metabolism in plants and animals. This enzyme catalyzes the hydrolysis of the terminal peptide link at the carboxyl end of the peptide chain. The enzyme is selective and works only when the R group in the terminal amino acid is a aromatic or a branched aliphatic chain and has L configuration. Thank you.